It's really an honor for me to be here. I congratulate you on 100 years of really good entomology here at Purdue. You certainly have a rich history, and we've benefited at Ohio State. We recently, as many of you know, we hired Midapali. We hired Andy Michael a few years before that, and Luis Cañas. So we benefited by this fine education that you're giving your folks. So thank you for that. Well, I'm, as Steve alluded to, my lab has been interested in diapause and cold hardiness. And a few years ago, we were sitting around having coffee and said, you know, if we're really serious about this, we should go to someplace a little more extreme than Ohio and see how insects survive in that tough environment. And Antarctica seemed like a good place to go to do that. We were aware that there was an insect that lived there, a little midge called Belgica antarctica. So I wrote a grant proposal to the NSF Polar Program. I must say I'd never written a grant proposal for an insect I'd never seen before, and my biggest fear was we get funded and go down there and find out the insect was no longer there. But fortunately we did get funded, and we went down and the insect was still there, and I'll tell you a little bit about that today. In fact, my presentation is going to be sort of the first introduction of what it's like to live and work in Antarctica, with slides of one of my favorite places on the face of the earth, and then I'll talk a little bit about some of the research we're doing. So it's a lot of, you know, hey, here's Antarctica and here we go. Now, I gave a talk to my own department with this title some time ago, and our secretary thought she was saving my reputation by putting an S on the end of insect. She was sure I meant insects, and she advertised it as Antarctica and its insects. But no, there really is only one species of insect that's endemic to Antarctica. There have been a couple recently that have come into the tip of the Antarctic Peninsula that over from Tierra del Fuego. A couple other midge species are now living there, but this one that we'll talk about today is the only one that is endemic to the continent. Antarctica, you know, it's quite a bit larger than continental U.S. And as many of you probably know, there was a wonderful treaty signed in the 1950s during the Eisenhower era, I think 1954, the Antarctic Treaty, where the countries of the world signed on in agreement that Antarctica belongs to no one. It belongs to everyone. The only thing that can be done there is things for the betterment of humankind, and there can be no military activity there. There can be no exploitation of economic gain on the continent. So, hey, this is my kind of a continent. I like continents like that. So, as a result, many of the nations of the world have indeed established research stations on Antarctica to sort of claim a little bit of, you know, especially for research activity. Now, the U.S. has 3 stations: Palmer Station here in Antarctica, on the Antarctic Peninsula where I've worked, South Pole, and McMurdo. These are both much larger stations. In fact, McMurdo, it really goes up to, it's a little city of 1,000 people or so in the summertime. A lot of work there, a lot of geological, climatological sorts of activity in there. Palmer Station, by contrast, is a much smaller station, can only accommodate a maximum of about 40 people, and of those, a lot of the people are support staff. Only about 15 scientists can be there at one time, so it's a small station. To McMurdo and South Pole, you can go in, you can fly into those. They have airstrips and you can fly in. You cannot fly to Palmer Station. You have to go by ship. So we fly down to Punta Arenas, Chile, the tip of Chile, get outfitted with our extreme weather gear, and then board the NSF's icebreaker, the Lawrence Gould, to make the journey across to Palmer Station. Now, of course, one of the other things, since it's so inaccessible, you have to plan your experiments very well in advance. You know, we're used to, out of a chemical, you know, order it the next day we have it. That doesn't happen there. If you don't go with absolutely everything you need, you don't get it. And Raytheon Corporation has the contract for getting what we need and our supplies. And actually, the first year we went down, we had on our list Trizol as one of the things we needed, because we needed to we did trizol, and all the other molecular things we would have done, we just couldn't have done without trizol. We got to Punta Arenas and found out that the trizol they had expired. So they flew a plane back to Santiago to get us a bottle of trizol and bring it back again. So it had to be the most expensive bottle of trizol on the face of the earth. But they do a good job of supplying those sorts of things for us. So NSF runs this icebreaker. This was actually our first year down. Two of my postdocs, Joe Reinhardt and Scott Hayward, shown here, out starting to head toward Antarctica. Now we're there, but what I don't tell you about is the time when I was not in a position to take any pictures. That it's a journey of about 4.5, 5 days, depending on the weather. And for about a day and a half, 2 days, you're in the Drake Passage, from the tip of South America until you get to the Antarctic Peninsula. And the water just moves around there in really high waves. 20-foot waves are routine. 30 to 40-foot waves are not at all unusual. And you really wonder why on earth you're doing this. And when you get there, you understand why. But there's a time in there that the cook is not terribly busy, and you're strapped into your bunk and suitcases are flying all around, and it's a pretty crazy time. And I mean, the only thing that, I can't imagine the ship survived, but it's made the trip many times. That was the only thing that I could say, okay, it's made it before, so I guess it'll make it again. In fact, one of my postdocs this last time was hooked up to an IV the whole time back, so it can be a rather traumatic journey to get there. But once you get there, it's worth it. So this is a station, not much of a place, just out on a little barren rock on the peninsula, and smallest, as I said, smallest U.S. Antarctic base. And this is a little closer up, the main building. We sleep up here in the top floor. You go down a flight of stairs, have your breakfast, go down another flight of stairs to your laboratory. So how convenient could that be? The boathouse here, where the Zodiacs that we use to get out to the islands. This is the picture of the lab, actually quite well-equipped labs. We, again, tell them what we want in advance. This is not part of the grant. They provide all the supplies and equipment that we need for our experiments, and just do a really good job of keeping things available and making things available. And this is why we went, to study the largest terrestrial animal that lives in Antarctica year-round. Okay, there are seals and penguins that are a lot bigger, but they just come up on land for a couple months or so. This is the largest animal that lives on the land year-round, the larvae of the midge, Belgica antarctica. It takes it a couple years to get through its life cycle. It only can be active during the Antarctic summer. So we've been going there normally right after Christmas and stay until mid-February or so. That's the active period for this. So during that time, it's out feeding on bacteria and some algae and things like that. Then when it gets cold again, it freezes over. These guys are encased in ice then for 8 or 9 months out of the year until it thaws out again and they go on their merry way once again. The adults come out in about early January. This is a mating pair, a male, much smaller than the large female. You see they're wingless midges, as indeed in many of these high windswept islands. You're not going to want to have wings, or you'll be cast out to sea fairly quickly. So they're wingless. They come out. They don't feed. They simply come out, mate, lay eggs, and die. They're dead within 10 days after emergence. So they don't have much of a life. So things that we've needed to do in the adults, we've needed to do right away as soon as we get there. So the midge does not live actually at Palmer Station. Palmer Station is right here. But you see a whole bunch of little islands out in the ocean, and that's where we need to go to find the midge larvae. These are islands that are accessible through Zodiac. This is one of my graduate students, Nick Tietz, who's captaining our ship that day. So we go out in these little Zodiacs and work our way through the ice. There's the Zodiac Park. We keep in radio contact with the main station. We're supposed to call back every 20 minutes or so to let them know we're still okay. And the weather can change so quickly there. From one time when there's ice like this, and the next 20 minutes later it might all be gone and very clear. Or ice packs may come in that would prevent our getting back to land. And most of the islands do have provisions, caches there, where if you have to stay overnight, you could do it. But we've never been caught, and we've been able to go back and forth in these islands. There's sort of about a 5-mile a radius that we're allowed to go out in these Zodiacs. So we have to take boating lessons when we first go there. And it's sort of interesting. The boating instructor the first year told us that if you go overboard, put your hand like this, so the hand freezes in that position so they can grab you. And okay, well, what have I gotten myself in for? So some little tips like that. Should you ever find yourself in a freezing condition like that, remember to do that. So this is the sort of habitat where we find the midge. This is one of my former postdocs, Joe Reinhardt. You see the green Prassiola here. That's an algae that's very common to that area. And we usually find the midges sort of in the periphery of the penguin rookeries and near the elephant seal wallows. And we think a lot of that nitrogen runoff from the penguin rookeries and from these elephant seal wallows are really important for nurturing the bacteria that these midges live on. Now this is really a very special area, much like the Galapagos. The animals are not at all afraid of you there. I mean, we're there working and the penguins will waddle over and look at what we're doing, and the elephant seals come sort of sliding over to check us out. And it's really a very special environment in that regard, just much like the Galapagos in that regard. So the islands each have a little bit different character. This one is one with a lot of moss on. Again, my postdocs Scott and Joe Reinhardt. This is Rick Lee, longtime collaborator. Rick is at Miami University in Ohio. He and I have shared grants on this project since the beginning, and have done a lot of work together. And this include this picture just to remind me to say this is a high school teacher, Luke Sandro, that we took along the first year. And we've taken a teacher with us every year. We've been taking down a team of 5, and one of those people has been a teacher. First year was Luke, who was a high school biology teacher. We've taken an elementary teacher, a middle school teacher, and we've a range of teachers through the years. And as NSF proposals these days, you're always supposed to talk about the broader impacts and the outreach sorts of things. So our teacher is sort of our outreach program, and they're in connection with the schools around Ohio and around the nation, really, who've been following the website, and some of them have been very engaged in interactions at a really impressive level with students. So they've been an important part of our team. We use them, not engage them in the research as well as much as we can when it's appropriate. Okay, just some great icebergs, as you might imagine, and calving back here. This is from Palmer Station looking across Mount William near the Palmer Station. That's again, my graduate student Nick Tietz there. Another sort of habitat that's very characteristic of Antarctica. Not much substrate, a lot of rock. I include this mainly because from Palmer Station up, it just looks like it'd be really fun to just walk and keep going, but it's really not safe to do that. They have limits on how far we're allowed to go because of some crevasses and the danger of falling in those. So we're limited somewhat in how far we can go because of that. People come to visit us while we're there. We've had a number of ships, especially there in the summertime, a number of sailboats like this one who've come down to visit. And the small ships, there are a lot of, Antarctica has been a really popular place to visit recently, so a lot of the, National Geographic Explorer, the Endeavour, and those ships have come. Ships that have, say, 100, 115 people on it, they're allowed to stop at the station and come visit the station. We'll serve them brownies and coffee and sell them T-shirts and all those kinds of wonderful things. So they can do that. But of course, the larger ships are just much too big, and they do not, can't do that. So this is one of the, I think this is a Rotterdam, one of those dam ships that has huge numbers of people on it. And as part of our outreach there, we're asked to go out and talk to these people and go up in their theaters, and of course they have these wonderful buffets that we enjoy going out to do, and they'll often send cheesecakes back with us. But the best thing are all the freshies they send back with us, the fruits and the vegetables, because after soon after you arrive, you soon deplete the fresh fruit and you're left with the dried things. So it's kind of fun to have these ships come by and drop off some good food for us. But this is frankly among the scarier things you do. We're going out in these little Zodiacs, and when the water's rough, at least, you have to catch it just right. They throw this rope ladder down over there. We have to grab it just the right time and then go up and give our talks to these people. So it's kind of an interesting experience. Okay, well, the midge isn't the only insect, the only arthropod that's there. This is a tick, Ixodes uri, the seabird tick that comes up and feeds on the penguins when they first come into the rookeries. So, and then you can find these in pretty large numbers under the rocks. They come in and feed right away, and then they go back after a feeding of about a week or so, go back and crawl into the same rocks and hang out there. What's particularly interesting about this tick is that it has a bipolar distribution. It's down in Antarctica. It's also up in the Arctic, but nowhere in between. So it must have caught a ride in one of those bipolar birds like the Arctic tern or the skua that goes back and forth, and at some point got established on both ends of the world, but not between. So, and apparently, I mean, people have looked at it pretty closely. They just cannot tell any difference between the ones in north and the ones that are south. There are also a number of little mites. This is a little black mite that you often find in pretty good numbers. We've done some work with both the ticks and the mites, as well as the colembula shown here. There are several species of colembula that are there, about 3 species that are pretty common in the area, and we've done some work on colembula as well, just because we were there and had time to look at them too. Well, okay, the elephant seals, yeah, you can't help but like them, and their big eyes. And there's one coming up to our lab. That's our lab door, checking us out close up. Leopard seal, got to be a little more careful for them, especially when you're out in one of these air-filled Zodiacs, just a little rubber raft sort of thing, and they'll come up on one side of the Zodiac and look at you and disappear, and then come up on the other side and look at you, and in spite of their smile, they, you do have to be careful for them. I have seen where they've taken a bite out of a Zodiac, and they do indeed deflate. Now, the good thing about Zodiacs is they have 4 of these inflatable panels, so if they bite one, you can still stay afloat for a while at any rate until you call back for help. But we try to give them a reasonably wide berth. Actually, there was a British graduate student who was killed by a leopard seal a few years ago at the Rotherham station nearby Palmer Station. Fur seals. And lots of humpbacks. We're out in our Zodiacs going to these islands, and there's a humpback, and they sometimes come up and check us out in a wonderful way too. That's a little bit too close. There's a couple of one of my graduate students, Josh Benoit, and one of Rick Lee's graduate students, Michael Nitsky, in that Zodiac. We were over in this one, and the whale came up between us, and that was, a little too close. Delhi penguins, that's a dominant penguin in that area. Their numbers are going down quite a bit. but still respectable sized colonies here. Now this, I don't quite get this. I mean, late in the season, this is a chick that they always sit like this. The mother sits on their head, so they can't, I guess they can't see what's going on, but it looks a little ridiculous for the parents that are, the chick seems larger than the parent, but they take that position, which is kind of interesting. So actually one of the reasons the leopard seals are so abundant, especially in late February, is this is about the time when the chicks are about ready to take to the water for the first time. So leopard seals come in and have a feast as these young penguins head out. These are chinstrap penguins. Actually, the first years down there, didn't see any of these, but with global warming occurring, this is a little warmer area penguins, so they're moving into this area, as are the Gentoos, which are recently moving into this area from further north. Sheathbills, or the chickens of Antarctica. Skewers, which are really nasty birds. They'll dive bomb you, and really not a nice bird at all. Giant petrels, great birds. These are long-lived birds that can live 50, 60 years. And some of the researchers who've been working in those, Bill Fraser's group, spend a lot of time just getting those birds familiar with them. And they would come in and first years just sort of stand by them. And they're now at the point where, you know, the petrels come back to the same nest year after year, so the same individuals are coming to the same place, and now they can just go in and reach under, pull the chick out, weigh it, and do what they need to do, and the petrel does not mind at all. They allow them to do that. So really, you know, these are awesome, you know, 6-foot wingspan birds. Antarctic terns as well. Okay, blue-eyed shag, a cormorant, it's pretty abundant down there. And now, sort of, what do we do when we go there? Well, this, just to introduce that, here's a couple little tubes being chilled in 10,000-year-old ice, a bar ice. Well, we did an ice machine down there, but postdocs just thought it was cooler to chill their tubes in ice that was really old. So that's just to introduce that. And just let me tell you a little bit about some of the kinds of experiments we've been doing there. We've been, a number of years ago, we described something we refer to as rapid cold hardening for temperate species. That represents, say, at 25 degrees, or flesh flies, or any other insect really. Hold them at 25 degrees, take them down to -10, they're dead. But if you take them at 25, put them to 0 degrees for as short as 10 or 20 minutes, or here is an hour, and then put them to -10, they survive very nicely. So there's some really rapid response that is enabling them to then survive at low temperature. Well, we tested to see if this same sort of thing would happen in a frozen midge, and indeed it seems to happen. In this case, we're looking at survival. This is just -10 at 24 degrees. 24 hours, they can handle that nicely. -15 for 24 hours. You can see they don't, about half of them are killed. But if you give them first 1 hour at -5 and then put them to -15, they do very well. And here, -20, none of them survived direct exposure to -20, but -5 for 1 hour before -20, and they survive very nicely. So here's the animal actually in a frozen state, but is still able to make that some kind of very rapid adjustment to low temperature. This just shows that some tissues are more vulnerable than others. This is let's just look at the fat body data. Fat body control at 4 degrees. 4 degrees is pretty much the air temperature during the summer down there. And here, if they're frozen with no prior treatment, just immediately to -20, you see the red indicates death. Green is healthy cells. Red is that. Here, if you give them this 1 hour at -5, you can see that the cells are very happy. So some of the other tissues are less affected, but the fat body is particularly vulnerable to injury and survives after rapid cold hardening. We think there's an important role for calcium in this. I won't go into big detail here, but we're looking here at just cell viability. And here we use BAPTA and some other agents that can block the calcium. The black lines indicate when calcium is present, the cells survive very nicely. But if you block the calcium, you cut down in survival. So we think that the calcium there's an important role for calcium in this rapid cold hardening response as well. Well, this slide just of ice crystals, just to remind me to say that, of course, the real danger with ice, with low temperature, is the formation of ice within the cell. You know, ice crystals form, the cell ruptures simply by expansion and the little points of the ice that are a problem. So what do you do about this? And these larvae have a good solution to that. And that is they are able to get rid of their water very effectively. Looking at percent water loss, this is across days, these are a couple different humidities. You can see they can lose their water really pretty quickly. Well, most insects can tolerate maybe losing 20 to 30% of their water. These guys can survive a loss of 70% of their water. They look like dried up little raisins, and you can't imagine they're still alive. But you pour water on them, they plump up and go on their merry way. So what we see here is that just after 2 days at this humidity, lose 30% of the water. And now look at survival in response to -10. Those that were fully hydrated died right away, but if you lost 30% of their water, they could then survive that low temperature. So one of the important strategies they seem to use is get rid of your water, and then you can survive at a lower temperature. So that seems to be an important adaptation for surviving in that environment. There's some other things that go on too. I mean, they increase their trailose levels. They increase glycerol too. But you know, I must say that isn't as impressive. We have a lot of temperate species that really boost their glycerol and trailose, and it's not. It's, you know, it's a bit of a change here, but it's not as dramatic as it is in many of the temperate region species. They also see when they get dry, they sort of clump together and form clumps like you see here as well. One of the other things that we came across was what we think is an important role for heat shock proteins in these larvae. Heat shock proteins, as I suspect many of you know, are involved in protecting the proteins from denaturation, and also, I mean, there are a number of different categories of heat shock proteins. HSP70 is particularly well known, very highly expressed in response to a lot of injury. There's a cognate 70 that's constitutively expressed. It's just around all the time. And there's HSP90, and then a whole bunch of these small heat shock proteins that, you know, normally as under heat conditions or conditions that would denature the protein. It could be either heat or cold or a number of other things. The protein denatures, and then you go on a bit further to a protein aggregate. But the heat shock proteins can operate in these various categories to either target these proteins that are being denatured, or to prevent, or also to target some for denaturation. I mean, what seems to happen in an early stage of denaturation, this cognate 70 comes in and identifies the protein HSP23, one of the small heat shock proteins, latches on to. A little bit later, the cognate drops off and HSP70 comes in and has a role in refolding the proteins and preventing the breakdown or targeting some for removal. Well, we had shown a number of years ago that during diapause here in the winter, many insects are making heat shock proteins all the time too. And so we wanted to test that something like that might be happening in Antarctica in these midges. And indeed that appears to be the case for the larvae. These are just several different heat shock proteins. Let's look first here at the adult. The adults give what we would say is a very normal heat shock response. At 4 degrees, the normal environmental temperature, no heat shock proteins produced. At a high temperature for an hour, 30 degrees, this heat shock protein response comes on very robustly, and then go up a little bit higher when it starts to kill them, it knocks off again. And that's true for HSP70 as well as HSP90. The larvae, though, it's a bit different. Here, even at 4 degrees, the normal outside temperature, these heat shock proteins are on. They're just making those heat shock proteins all the time, and they're continuously on. After you get up to a certain high temperature, they drop off, but the heat shock proteins are on really all the time, and protecting, we think, playing an important role, perhaps in maintaining the integrity of some of the enzymes that need to function during survival at low temperatures. So we have this parallel of here's an animal living in a very cold environment, making heat shock proteins all the time, and our wintering species here that are doing a similar thing. What was that? Okay. Well, another group of proteins and genes that we've looked at are those that are involved in oxidative stress. This we thought might be an interesting area to look at, some of the catalases and SODs. And indeed, you can see, I mean, there's really a lot of high UV component to that Antarctic sunlight. And much like the heat shock proteins, here at, even without any sunlight exposure, SOD and catalase are very highly expressed. So much like the heat shock proteins, these are genes that are just on all the time in these Antarctic midges. These are midges that are able to survive a whole range of environmental stresses, from reasonably high temperature to low temperature to dehydration. And here in seawater, we can stick them in seawater and they'll survive very nicely for at least a week. And they can survive in fresh water as well. You see a number of different responses that occur in seawater. They decrease body content and a number of things here that we've noted. So, and it's indeed, these are realistic conditions because they're living close to the sea. Sometimes storms come by, as is happening here, and bring salt water in over their habitat. Other times in the spring, when the ice melts, they're in a pool of fresh water. So they're able to handle all kinds of rather extremes, and the high nitrogen content of the wash from the penguin rookeries. Just a lot of interesting extremes. Well, we've gone after the sequencing of this gene using for genomic sequencing as well as RNA-seq, with particularly a goal of trying to see what are these responses that occur in relation to dehydration, since we think dehydration is so important. We're in the process of trying to wrap up and get a couple papers out on this work fairly soon. And I'll just share a few things that are happening here in response to this dehydration response. We see a number of genes that are associated with autophagy that are highly upregulated, whereas apoptosis is shut off. So this process of programmed cell death is shut off, but autophagy, this recycling of nutrients within the cell, genes associated with that process are coming on very strongly during this time. And just another example of things that are shut down, in this case, glycolysis and TCA cycle, you shut down glycolysis, you shut down activity within the TCA cycles. There's a lot of consistency here within the sorts of major pathways that are being turned on or off in response to desiccation. We've done some metabolomics experiments too, which look at some of the several of the compounds involved in the TCA cycle, and you can see in response to desiccation, we have a couple different kinds of desiccation we do, but you can see here several major players in the TCA cycle are shut down in response to desiccation. Citrate goes up. It looks like, in the TCA cycle, citrate just accumulates there without being going further into the TCA cycle. And some of the key genes involved in proline is one of the components that comes up nicely during dehydration. Here shows the protein levels that are high, proline levels that are high in desiccation, and here the gene, one of the major genes that leads up the proline synthesis, is nicely turned on. So we're getting this kind of nice consistency between the transcript data and the metabolomics data, and in most of our work. Another set of genes that we've been quite interested in, or proteins that we're interested in, are the aquaporins. Since release of water, getting rid of water is really important, we thought we'd go after the aquaporins. These are, as I'm sure many of you know, the pore proteins that the water goes through and can be really important in water transport across the cell membrane. And we have actually the beauty of the genome, having the genome now, we worked for a long time to get one of those aquaporins, and we reported on it, and we tried really hard to, we knew there must be others, but we weren't not able to get them, and now they have a genome. We suddenly have, 5 other aquaporins that we can play with, and of course that's opened up a lot of new possibilities, as all of you who are working with genomic projects know what opportunity this gives you. So this particular aquaporin drip comes on strongly in response to some dehydration responses, and we're in the process of teasing out all these different aquaporins. What does it mean to have so many of them, and are some of them being regulated by different forms of dehydration? Or are they present in different tissues, and what is the meaning behind all those different dehydration responses? Well, using a proteomics approach, when we look at what these midges do in response to dehydration, you see a whole bunch of, the vast majority of these proteins that are different are involved in contractile proteins, parts of the cytoskeleton. And as you well might imagine, when an insect is dehydrating like that, your body is making all kinds of changes in form and shape as you become smaller, and we see, looking at the protein level, just lots and lots of differences in the proteins that are involved in those kinds of responses. Well, the final little bit of data I want to talk about is switching over to the clock genes. I mean, here we have a situation in Antarctica where you go from the most extreme in day length, from essentially continuous day to continuous dark. Well, do you need clock genes in that kind of environment? And if so, what do you do with these? And we've cloned all of these clock genes from Belgica, Antarctica now, and we're in the process of trying to understand what they, how they work in this kind of an environment. And indeed, as many of you probably know, I mean, this gene period and timeless in particular have been very well studied, and they characteristically, you look at a light-dark cycle across 24 hours, you see routinely a peak that goes up, reaches a peak around the time lights off, and then goes down like this. Well, you can see these guys the gene is present, they're making the protein too, but they're certainly not cycling. These things are flat. All of these clock, there's maybe a little, I think that's a straight line too. The others are really conspicuously straight lines. So they're very, very flat. Although the clock gene is there, it's just not cycling the way we might have expected. And this is some experiments, actually field-based experiments, looking at what the clock gene is doing in the field. And you can see here, this is under a condition where there's about 3 hours of darkness, but by darkness, it just means the sun went just down under the horizon. You could still read a paper or anything at night, and so it's not really dark. It's much brighter than it is at moonlight, full moonlight here. So it wasn't really dark, but yet the sun was below the horizon for a couple hours. You can see the clock genes, again, under natural conditions, field collected samples, just not showing any periodicity as well. And then we looked at the locomotor activity of both the larvae and adults. This is just some results from the larvae. And we had cameras down on them and looking at recording their activity. And you can see they're just going flat out all the time. There's no indication of any time of the day which they are less active. They're just go for it. They're around, I see it as sort of an opportunistic situation where you only have a few weeks to do your thing, so you just go flat out for the whole time that you're doing that. So we think that that's probably what is happening. Well, by the time we normally have been leaving, mid-February or so, the ship comes back to pick us up. And the first signs of winter are starting to appear. we have been able to take midges back home with us. We take thousands of them back with us, then bring them home, rear them in my cold room at home at 4 degrees. They're very happy there. You bring them out to the room temperature. They're dead in in 12 hours or so if you bring them out to room temperature, but as long as you keep them in the cold room, they're very happy, and we maintain a low light intensity on them there, and we have not been able to rear them. I mean, we get adults coming out sometimes. We don't think we have ever succeeded in getting them to deposit eggs and go on to another generation. But at least we can bring them back and say in 1 February, we'll still have larvae that we can use the following December or so. By that time, they start to become a little less, the quality is down a little bit. But it gives us a chance to extend our experimental season by 8 or 9 months by doing that. So I think this slide just sort of summarizes some of the things, the lessons we've learned from Belgica and how you can survive there, and some of the tricks that this species engages in. Just get rid of your body water, and of course it's no small feat to be able to do that. A lot of insects do not have the capacity to do that, but they do and can still survive. So some neat lessons that can be learned there. And we think turning on the heat shock proteins is important. Turn on your antioxidant responses, and some metabolic changes that are probably quite important for it as well. And then boosting this recycling pathways, all that autophagy genes that are turned on as part of the dehydration response, and then shutting down glycolysis. And then finally, just be an opportunist. You don't need a clock when you live there. Just go full out all the time and go from there. So that I think is just the last slide. This project was supported by NSF's Polar Program. I don't know if we're working on a renewal application now. I don't know if we'll get to go again because there's so much pressure on people wanting to go there, and so limited number of people who can go at one time. So they may very well say we've had 2 rounds of funding and that's up, but hey, we do have the genome now, so it'd be a shame to shut us off at this point, but hey, who knows? They might very well do that. And obviously we're limited to, we can only work with the animals that we can get from there. Okay, a number of my graduate students have gone. The only current one is Nick Tietz, who went a couple times, but Josh Benoit and couple other graduate students, a couple postdocs. Elena Kovalkova was there this last year. Postdocs Joe Reinhardt and Scott Hayward went in previous years. Then again, point out Rick Lee at Miami University, a longtime collaborator in this project. Joanna Kelly is involved with us on the sequencing of Belgica right now. So I think that's my last slide, and thank you kindly for your attention.